Bohr's effect. It occurs during oxygen delivery at tissues. In lungs, oxygen combines with hemoglobin to form oxygenated hemoglobin. The RBC is transported to the tissue. At tissues, carbon dioxide diffuses inside the RBC. It combines with water molecule, and in presence of carbonic anhydrase, it forms H2CO3. It further dissociates into H plus ions and HCO3 minus ions. The oxygenated hemoglobin, in presence of H plus ion, it dissociates and releases oxygen molecule to the tissues, and it itself combined with the hemoglobin. So Bohr's effect says that hemoglobin's oxygen affinity is inversely proportional to the H plus ion and carbon dioxide concentration. So higher the H plus ion and high the CO2 concentration, the hemoglobin releases the oxygen to the tissues. Now we'll talk about Bohr's shift. It is the rightward shift of oxygen dissociation curve in presence of high CO2. We have the graph of oxygen dissociation curve. On x-axis, we have PO2 in kilopascal, which corresponds to the dissolved oxygen. On y-axis, we have percentage saturation of oxygen, which corresponds to the oxygen content of the blood. So at normal PCO2 level, the PO2 of 5.3 kilopascal, it corresponds to saturation of 75%. It is the normal venous oxygen saturation, and the difference between 100% and 75%, it corresponds to the oxygen delivery to the tissue. At high PCO2 levels, the PO2 of 5.3 kilopascal, it corresponds to the saturation of 50%. So the difference between 100% and 50% is higher. It means that more oxygen is delivered to the tissues. So Bohr's effect causes more oxygen delivery to the muscles and tissues. In nutshell, acidosis promotes unloading of oxygen at tissues. It increases oxygen delivery to the tissues, and that's why saturation drops significantly.